This is a recording of bee-friendly microbial control of arachnid and arthropod pests, presented at the 2019 American Beekeeper Federation Conference in Myrtle Beach, South Carolina. This presentation is showcasing the work of the small but hard-working team of Strong Microbials who are Jennifer, Vera, Rich, Slava, Dan, Carol, Earl, Nick, and Layton. You can see them all in the upper photo. And every day in Milwaukee, Wisconsin, we grow and blend microbial products, also grow our bees and the next generation of beekeepers. Strong Microbials is founded in scientific research, and our researcher Jennifer is posting the most interesting items that come out in the science news related to bee health and bee biology in her blog, beesciencenews.com. Even though we were growing microbes, we didn't raise bees until several years ago when the headlines, such as this one, "A World Without Bees," drew our attention and empathy. Motivated to start beekeeping, we also set out to help save the bees. In the course of this journey, we began by identifying the various problems threatening the bees today. As you all know, pests and infections such as varroa mites, small hive beetles, nosema, and chalk brood cause beekeepers economic and colony losses. Also, pesticide use is a problem for bees and beekeeping. Despite the existence of chemical-free methods to eradicate pests, Americans use more than 1 billion pounds of pesticides each year. The use of chemical pesticides increases every year in the US and in the world. So, two problems facing the bees now are pests and pesticides. Not only chemical pesticides are toxic to the bees, they are not always necessary for successful agriculture. Strong Microbials is implementing sustainable microbial agricultural solutions, which will ultimately lower agricultural pesticide use, restoring microbial soil diversity. As Jonathan Lundgren says, if you want to heal the bees, heal the soil. Yet as another scientist, Dr. Dennis van Engelsdorp says, of the pesticides and chemicals detected in the hive, majority are put into the hive by beekeepers themselves. Why is that? This includes antibiotics, many chemical treatments, fumigillin and fungicides, and antibiotics and antimicrobial chemicals are toxic to beneficial microorganisms. Consider this analogy. Just like pesticides are toxic to beneficial pollinators, antibiotics are toxic to beneficial microorganisms. Beekeepers, why do we treat our bees with antibiotics prophylactically? This needs to change. Let's try replacing chemicals and antibiotics with probiotics. Listen, pests and pesticides are the problems that have the same solution, and that's why farmers and beekeepers must change our reliance on pesticides and antibiotics, because pests and pesticides have the same solution. And the solution is microbes. Microbes are a part of healthy soil, a healthy gut, and are now used for beneficial microbial pest control. This image shows microbial pest control in action. Crop growers now are using fungus called Beauveria bassiana to control aphids on the underside of leaves. Microbial controls do not leave chemical residues or lead to resistant pests, nor do they affect pollinators. And despite more than 1 billion pounds of chemical pesticides used every year, the use of chemical-free methods to eradicate pests, indicated by the thin green sliver on the bottom of each of these black bars. It's increasing every year, and the main reason why the use of microbial products is growing is because they work. The war of the worlds, so to say, between different microorganisms made them fantastic at the production of chemical weapons or metabolites, at controlling when, in what combination, and how they are released upon their enemy. Microbes are also exquisite at building protective structures and manipulating their environment. This is an example of extracellular matrix produced by bacterium Bacillus subtilis in order to make the environment safe and hospitable for this organism. Chemical products such as antibiotics were discovered first as microbial metabolites. In 1928, discovery of antibiotic penicillin, a metabolite of Penicillium fungus, gave us the ability and knowledge how to treat deadly infections. In 1949, another antibiotic, fumagillin, which is a metabolite produced by Aspergillus fungus, was discovered and used as an antibiotic chemotherapy agent for humans. And then several decades later, fumagillin was called on to treat Nosema-infected honeybees. Strong Microbials continues microbial product discovery to help solve many of the honeybee problems. Since the introduction of our first probiotic product, Super DFM Honeybee, 5 years went by and we noted substantial benefits in treatment of chalk brood and Nosema infections. Today's presentation will focus on new products which we are developing. And the first project that I will tell you about is a product to control and stop hive beetles. The goal of this project was to identify a microbial isolate that can control small hive beetles, and after screening different microbials, we have pinpointed one that had efficacy in controlling small hive beetle. We're currently registering this product with EPA and wish to bring it to market this year. Our initial tests focused on the number of adult small hive beetles in every honeybee colony, and the isolate selected for BioBeetle successfully decreased the number of beetles very rapidly on a time scale of beekeepers also made an observation, which is captured here on video, that larvae of small hive beetles, not only the adult beetles, but the larvae were strongly repelled and killed by the microbial powder.
Okay, this is what happened when we dusted a little bit of the red group onto the frame. All of the larvae have moved out of the comb area and moved into the top of the frame area.
These are the larvae of the small hive beetle, and we raised beetles and larvae in the lab to confirm that exposure to the BioBeetle microbial product results in 100% mortality of the small hive beetle. This is another video of small hive beetle in the lab. On the left is the control group, which is alive and moving around and 100% viable, and on the right is the BioBeetle exposed larvae, which are 100% dead. Now, after finding the optimal concentration and application methods, field testing showed that BioBeetle was more effective than treatment with fipronil, which some beekeepers use in order to control beetles. All of the hives are cleared of infestation completely with BioBeetle compared to fipronil. The BioBeetle microbial control product will become available in 2019. And I will tell you about another microbial project seeking a control for varroa mites. In order to find a fungal or bacterial isolate that control varroa, we have turned to literature and screened different microorganisms, including Metarhizium and Vivaria and Burkholderia, to find one of these microorganisms that was selected for its efficacy in terms of topical toxicity to varroa mite. In this video, honeybee pupa, which are used to raise and study varroa mites in the lab, you can see that they're dead and the mites are alive, and you can see several mites moving around in this container. 2 of them, which are in the center of the video, are touched with a fine brush dipped in microbial solution. You can see that the mites are moving around and crawling around on the larva. And the two mites in the middle, on top of this larva, have been touched with the microbial solution. What we find is that such topical application of this microbial stuns the mite and kills it in minutes. Now the microbial solution application to the second mite, as you can see right now, also results in stunning and killing of this mite. You can see it's slowing down and stops moving in seconds after the application. Alright, so then we moved on to field applications and are now finalizing the best application method. So far we can stimulate significant mite drop with the microbial product, which is evidenced here by daily sticky board mite counts. Once we finalize the concentration of this microbial that gives us maximum efficacy, we anticipate the completion of these trials by 2020, and we will continue to bring different beneficial microorganisms for beekeeping. Please visit us at strongmicrobials.com.